Hi everyone, this is Glenda Ganzon and welcome to my Human Anatomy and Physiology class. And for today's video, I'm going to discuss about the cell differentiation. So stay tuned. There are a lot of questions like, how does a complex organism such as a human develop from a single cell, a fertilized egg, into a vast array of cell types such as nerve cells, muscle cells, and epithelial cells that characterize the adult? Throughout development and adulthood, the process of cellular differentiation leads cells to assume their final morphology and physiology. Differentiation is the process by which unspecialized cells become specialized to carry out distinct functions. So first of all, I would like to discuss about the stem cells. So what is a stem cell? A stem cell is an unspecialized cell that can divide without limit as needed and can, under specific conditions, differentiate into specialized cells. So stem cells are divided into several categories according to their potential differentiate. The first, embryonic cells that arise from the division of the zygote are ultimate stem cells, and these stem cells are described as totipotent because they have the potential to differentiate into any of the cells needed to enable an organism to grow and develop. So the embryonic cells that develop from totipotent stem cells are precursors to the fundamental tissue layers of the embryo are classified as pluripotent. So what is a pluripotent? A pluripotent stem cell is one that has the potential to differentiate into any type of human tissue but cannot support the full development of an organism. So these cells can become slightly more specialized and are referred to as the multipotent cells. So what is a multipotent stem cells? A multipotent stem cells has the potential to differentiate into different types of cells within a given cell lineage or a small number of lineages such as a red blood cell or white blood cell. And finally, multipotent cells can become further specialized oligopotent cells. Again, what is an oligopotent cell? An oligopotent stem cell is limited to becoming one of the few different cell types. In contrast, a unipotent cell is fully specialized and can only reproduce to generate more of its own specific cell type. So then, stem cells are unique in that they can also continually divide and regenerate new stem cells instead of further specializing. And there are different stem cells present at different stages of a human life. They include the embryonic stem cells of the embryo, fetal stem cells of the fetus, and adult stem cells in the adult. One type of adult stem cell is the epithelial stem cell, which gives rise to the keratinocytes and the multiple layers of epithelial cells in the epidermis of the skin. And adult bone marrow has three distinct types of stem cells: the hematopoietic stem cells or hematopoietic stem cells, which give rise to red blood cells, white blood cells, and platelets. Endothelial stem cells, on the other hand, which gives rise to the endothelial cell types that line blood and lymph vessels, and mesenchymal stem cells, which give rise to the different types of muscle cells. So this is a picture of hematopoiesis. The process of hematopoiesis involves the differentiation of multipotent cells into blood and immune cells. The multipotent hematopoietic stem cells give rise to many different cell types, including the cells of the immune system and red blood cells. So when a cell differentiates, meaning to say, becomes more specialized. It may undertake major changes in its size, shape, metabolic activity, and overall functions. Because all cells in the body, beginning with the fertilized egg, contain the same DNA. And how do the different cell types come to be so different? The answer is analogous to a movie script. So the different actors in a movie all read from the same script. However, they are each only reading their own part of the script. So similarly, all cells contain the same full complement of DNA, but each type of cell only reads the portion of DNA that are relevant to its own functions. So in biology, this is referred to as the unique genetic expression of each cell. And in order for a cell to differentiate into specialized form and function, it need only manipulate those genes, which is those proteins that will be expressed, and not those that will remain silent. So the primary mechanism by which genes are turned on or off is through transcription factors. And what is a transcription factor? A transcription factor is one of a class of proteins that bind to specific genes on the DNA molecule and either promote or inhibit their transcription. In this picture is a transcription factors regulate gene expression. While each body cell contains the organism's entire genome, different cells regulate gene expression with the use of various transcription factors. Transcription factors are proteins that affect the binding of RNA polymerase to a particular gene of the DNA molecule. So this ends my discussion about the cell differentiation, and if you have any question about this, please write it in the comment section down below and I'll be glad to answer them all. And once again, I would like to thank you all for watching this video and for listening to my discussion. Until my next video, bye everyone!